The following slides are going to go over the structures found in a neuron. The neuron depicted is a multipolar neuron, and multipolar neurons are motor neurons. Neurons consist of a cell body, the cell body being the biosynthetic and one of the receptor regions of the neuron. Biosynthetic region is the region that contains the nucleus and organelles like the nestle bodies that are involved in production of substances like the neurotransmitters. The neuron also consists of, in the case of multipolar neurons, several dendrites and one axon. The area where the body of the cell and the axon meet is the axon hillock. So the cell body has two functions. It is the biosynthetic region of the neuron, but it is also the receptive region, which means that other neurons could synapse at their body of the neuron. This will be an axosomatic synapse. The other region of the neuron are the dendrites. In the case of multipolar neurons, there are several dendrites that act as receptive regions. So neurons synapse with other neurons, and these synapses occur at two places. At the dendrite of the neurons, this will be axodendritic synapses. Other synapses take place between the axon of the presynaptic neuron and the body of the postsynaptic neuron. This will be axosomatic synapses. Keep in mind, most neurons are going to have a multitude of synapses at dendrites and at the cell body. The axon hillock is the place where the body connects with the axon of the neuron. The axon is a long appendage that ends at the axon terminals. The axon is the place that generates the nervous impulse or action potential, which starts at the axon hillock, and from there it is transmitted down the axon past the terminal branches into the axon terminals. At the axon terminal, in response to the neurotransmitter, in response to the action potential, the neurotransmitter will be secreted by the axon terminal region. So the axon terminal is the secretory region of the neuron that will secrete neurotransmitter in response to an action potential. In the case of a multipolar neuron, of a somatic motor neuron, the neurotransmitter secreted would be acetylcholine. Notice that this neuron is myelinated. The myelin sheath is a structure created by glial cells which wrap around the axon, creating an insulation that will allow the action potential or nervous impulse to travel rapidly down the axon. In between myelin sheath are regions that lack myelination called nodes of Ranvier. So the basic structure of a neuron is the cell body from which one or many dendrites come about, the axon hillock, which is the place where the body and the axon meet, and it is the area where the action potential starts. So this will be an area where voltage-gated channels will be found. Also coming from the body are the dendrites. Both the dendrites and the body or soma of the neuron are receptive regions, regions where other neurons may synapse. Secreting neurotransmitters that will be received by receptors at the dendrite or at the body of the neuron. The receptor regions contain channels that can receive the neurotransmitter, so will be chemical channels, chemically gated channels. The stimulus then creates a graded potential that travels to the axon hillock, where voltage-gated channels will initiate an action potential. The action potential travels down the axon to the branches, terminal branches or telodendria, that end at axon terminals. The axon terminals are the place where the neurotransmitter will be secreted in response to the action potential. The next neuron or the effector organ will, the receptor region will bind to the neurotransmitter. Remember that somatic motor neurons are myelinated, which means that myelin sheath will wrap around the axon of the neuron, insulating it and allowing for faster transmission of the action potential. The areas in between the axon, the myelination, where there is no myelination, are called nodes of Ranvier. The next material looks at myelinated axons and the difference in myelination between the peripheral nervous system and the central nervous system. Remember that in the peripheral nervous system, the neurons that myelinate axons are called Schwann cells. In the CNS, the neurons that myelinate axons are called oligodendrocytes. Notice that these cells work in a very different manner. In Schwann cells, one Schwann cell wraps around a portion of the axon, which means that several Schwann cells will be needed to create the myelin sheath along an axon. Oligodendrocytes, on the other hand, wrap around several axons. Notice also that not all axons are myelinated. As a matter of fact, most axons are non-myelinated. In the PNS, unmyelinated axons are still wrapped by Schwann cells. But in this case, several axons will be wrapped around by one Schwann cell in a loose manner. The reason why all axons in the PNS need to have a relationship with Schwann cells is because Schwann cells are also involved in repair of axons and protection of the axons. So this is an unmyelinated axon. The axon above is myelinated. When it comes to tissues in the nervous system, there are two distinct types of tissues that can be seen during a dissection. One type of tissue is called white matter, and white matter consists of myelinated axons. The other type of tissue is called gray matter. Gray matter consists of bodies of neurons and unmyelinated axons. White matter is called white because myelination is due to the cell membrane of Schwann cells or oligodendrocytes wrapping around axons. Cell membranes are rich in cholesterol, which is a white fatty substance, and that's what gives the color to those areas, the areas of the nervous system that house myelinated axons. In the central nervous system, gray matter is found in the cortex of the cerebrum, and it is also found deep in the brain in areas called nuclei. White matter is found below the cortex of the cerebrum and is organized into what is called tracts. In the spinal cord, and this is a cross-section of a spinal cord, white matter surrounds gray matter. Gray matter in the spinal cord are called gray horns, while the white matter is also organized into tracts called white columns. Remember, the gray matter contains bodies of neurons and unmyelinated axons, while white matter contains myelinated axons. So you could imagine bodies of neurons clustered, millions of them clustered in the cortex of the cerebrum, while their axons travel as tracks, within tracks, in the white matter. Bodies of neurons in the nuclei of the cerebrum and axons traveling through the white matter. Similarly, in the spinal cord, bodies of neurons will be found in the gray matter, while the axons are found in the white matter. Okay. In the PNS, peripheral nervous system, bodies of neurons are found in areas called ganglia. So this will be gray matter. While white matter are found in nerves. This picture shows you an actual specimen. This is the spinal cord. These are ganglia, or little bodies of gray matter that are housing the bodies of the neurons, while the axons travel through the white matter or nerves. So to summarize, white matter consists of myelinated axons. While gray matter consists of bodies of neurons and unmyelinated axons. In the brain, white matter is found in tracts. Gray matter is found in the cortex and in nuclei. In the spinal cord, white matter is found in tracts called white columns. Gray matter is found in gray horns. In the PNS, white matter is found in nerves. Gray matter is found in structures called ganglia.